Welcome to week 6 of high school biology. I hope that you guys are all having a great week. I just want to make sure that everyone can hear me. Uh-oh, we're terrified. There's nothing to be nervous about. We are going to go through every single step of dissecting a worm. Worms are fairly simple to dissect. There's nothing too gross about them. So do not be nervous. I am going to warn you guys that I am doing this class outside because it does smell a little. So if there is some sort of noise in the background, just let me know, and I will do my best to stop whatever it may be. So here we are in week 6 of high school biology. And actually, before we start with our lab, which we are going to start off right away because it may take most of the class. Oh, we have a pig. But none of you did the pig. Robert did the pig to share. So if Robert jumps in a second, he's going to share his project first, and then we are going to go ahead and do the dissection. But what we will do in the meantime is thank you all very much for going in and completing the earthworm dissection video for me, or the web link. That was just to give you guys an overview of what is going to happen today. It's nice sometimes to see it, especially if you've never done a dissection before you actually are doing the dissection. So if you haven't opened your lab kit yet, we will go ahead and do that in a minute. I have everything, I think, gathered here. It's not a big deal if you don't have some live earthworms. I do have a live earthworm. I have 3 of them. Hopefully one of them will cooperate with us. And we will go ahead and do that part of the lab I will share with you. If not, we'll skip that part. Sometimes the worms cooperate. Sometimes they have, they have other things on their mind. So before we do get started with our dissection, look at your whiteboard. On your whiteboard right now, I have a picture of an earthworm. There are lots of different species of earthworms. Depending on where you live, you are going to have different species. What I'd like to do is I'd like to open up the whiteboard to you guys. And remember, you can use the pencil to write, or you can use the black letter A to type. I'd like you guys to write some things that you know about earthworms. There are things that you have learned from reading the pre-lab, or things that you just know. What do you know about earthworms? Go ahead and put some things up here. And I just saw Robert pop in. So Robert, once we do this, then we're going to go ahead and talk about your pig dissection before we dissect our earthworm. So anything you know about earthworms, go ahead and put them up on the whiteboard. Okay, anyone have anything else? Throw it up here, please. Anything you know. I see two things up here. We have the pharynx gizzard crop. They have a dorsal side and a ventral side. They need to be in moist places so they do not suffocate. All right, and what else do we know about earthworms? Earthworms have an anterior and a posterior. They have a mouth and an anus. They also have a dorsal and a ventral side. They eat dirt, and through their digestive system, they are able to get nutrients from the soil. They are helpful to farmers because they create holes that provide oxygen for the plants. Those are both very good facts about earthworms. There are some other neat facts that I want to share with you guys. The first one being that there are over 6,000 species of earthworms worldwide. The one that you probably were able to connect, I'm just going to shut my video off for a second. The one you were probably able to collect, if you did collect them, is called a night crawler. It typically lives in the garden soil around houses. Sometimes when it's raining, these worms will leave the soil once it becomes waterlogged. There are over 180 earthworm species that are found in the U.S. and Canada. 60 of them are invasive, including the night crawler, which is probably the one you found in your garden. Earthworms lack lungs or other specialized respiratory organs. They breathe through their skin. So as we are dissecting, we are not going to see any lungs today. The skin secretes a fluid that makes moving through the underground burrows easier and helps keep the skin moist. There is an Australian earthworm that can actually shoot the fluid as far as 12 inches through their skin pores, so that is pretty gross. Each earthworm has both male and female parts and can produce eggs and sperm. Hopefully we will be able to see this in your earthworms today. At least one of us can find both. Baby worms emerge from tiny eggs. They are small, but they are fully formed when they emerge. They grow sex organs within the first 2 or 3 months of their life, and they reach their full size in about a year. Anyone have a guess as to how long a typical earthworm lives? Go ahead and either raise your hand or put it in the text chat. How long does a typical earthworm live? Not a worm, quote unquote, in captivity, but a worm in the wild. How long do they typically live? All right, we have a couple guesses. A typical earthworm lives 1 to 2 years, mostly because of predators. But they can live and have lived up to 8 years. So that's the answer to that one. Typically, an earthworm can range anywhere from a ½ an inch long to 14 inches in the U.S. However, in the tropics, they can grow much longer. Anyone have a guess as to how long an earthworm can be in the tropics? Actually, you are all way off. An earthworm in the tropics can be up to 10 feet long. Almost more like a snake than what I would think of as a worm. And to me, that is totally gross. I do not want to see a 10-foot-long worm anywhere. Okay, another couple of facts. During the northern U.S. and in Canada, most earthworms were eradicated or wiped out during the Ice Age. So most of the earthworms that we find in the northern half of the U.S. and in Canada are actually invasive species. Most of the natural ones have been decimated, their populations, because of the glaciers. And they actually are very beneficial to farmers in farmland or in your home garden. But they can be very harmful to forests. Northern forests evolved after glaciers retreated. So this type of ecosystem doesn't benefit from earthworms. The forest, like where I live in Connecticut, requires a deep layer of slowly decomposing leaves and other organic matter called duff that lays on top of the soil and basically fertilizes the soil as it decomposes. When earthworms get into the soil, they quickly eat up this duff, decaying leaves, and the nutrients become less available to young, growing plants in the soil. So instead of aerating and loosening the soil like earthworms do in a farmer's field, the soil actually becomes compacted. Because the soil becomes more compacted, some trees are not as successful. Sugar maples are a tree that has a very hard time in compact soil, so they do not benefit from earthworms because the earthworms eat all the decaying matter, so the soil does not get softer. All right, so yes, Robert, that's certainly a 10-foot earthworm is too big to put on your fishing hook. However, night crawlers are the perfect size to go on your fishing hook. So that's a little bit of background information about earthworms. Now we're going to jump to a little bit of background information about dissecting. After we do this, Robert is going to show us his pig dissection so you guys can get a glimpse of that. And then we are going to get started with our live earthworm and move to our dissection. Actually, you know what? I think we're going to have Robert show us his pig first, and then we'll switch back to the earthworm. So, Robert, I have gone ahead and uploaded your pictures to the PowerPoint screen. They probably are not in the order that you are going to talk about them. I think this is where you probably will start. So, Robert, why don't you go ahead and turn your mic on. The rest of you look at your PowerPoint screen. And this was the lab that Robert did for homework. I believe it was week 3. So, Robert, whenever you are ready, go ahead. Robert, I'm not able to hear you, so let's double check that your microphone is on. Go ahead to the bottom left-hand corner of your screen and look at the wrench. Click volume or microphone and make sure that your microphone, the correct microphone, is on and you do not have it muted. So let's go ahead and try that again. Unfortunately, I'm still not able to hear you. Can you just double check that? If you go down to the bottom of your screen, your microphone is not muted. If you use a speaker thing, sometimes your microphone will come up. Is anyone else able to hear? Robert, if you can hear him, just go ahead and write that in the text chat for me. Okay, so it's not me. Go ahead and check that, Robert, and we'll give you one more try. Otherwise, I will try to go ahead and explain your lab to everyone. Maybe, Robert, log out and log back in. Sometimes when you do that, for whatever reason, the connection is better. So we're going to give Robert a second to log out and log back in, and then he can go ahead and try again. The rest of you, why don't you make sure you have all of your lab supplies with you. If you don't have them, make sure you have some paper towels. Robert, once you're back in, go ahead. Nope, don't get the worm out yet. We're going to do the live part first. So we're just going to give Robert one more second to get back in here. It's really not that gross, Sarah. I promise. All right, Robert, go ahead and give it a try again.
Can you guys hear me? Okay. So first, I got the pig out, and my mom got some cardboard out, and we wrapped that in tin foil, and we put the pig on it. And my mom has a scalpel in her bathroom. So we started dissecting the pig, and we opened it up, and you could see all of its internal organs, and its heart, and its intestines, and lungs, and liver, and all that good stuff. And so then we started making little pins. We had little pins, and we put little names on them, the parts of the body, and we labeled the parts and put them in the pig. And then my mom and I wanted to have some fun, so we cut out some of the parts to get a better look at them. Show you a picture. And there's the pig with all the parts cut out of it. And my mom, being a nurse that worked in the ER, loves this stuff, so she wanted to go further. I don't know if you guys got pictures on there, but we cut open the skull and there was we got to look at the brain, and they cut out, yeah, there's the brain, and then we cut out the eye and got to look at part of the eyes too. I don't know if we have a picture of the eye on there or not. So yeah, that's the pig dissection. It's pretty fun, and my brother almost threw up. Okay, so there's no eye. But yeah, that was my dissection, and I hope you guys enjoyed that.
All right, great job, Robert. Oh. All right, great job, Robert. And I will be completely honest with you guys. The pig dissection is definitely the most, I don't want to say gross, but if you were going to be grossed out about one of them, it is certainly the one that you would get grossed out about. So it really wasn't that bad, I'm sure, Robert. Seems like he enjoyed it. And they actually went a little farther with the brain, so that was neat. I'm glad that you enjoyed it. So what we are going to do today is we are going to do lab number 22 in your lab book, which is on page 110. There are two parts to this lab. The first part is with a live worm, and we are not dissecting the live worm. We are just going to look at some worm behaviors. And then the second part of the lab is the actual dissection. Now we will do the first part first. Do not take your dissected worm out yet. You do not need it. If at any point you feel like you need to just step away because it smells or for whatever reason, that is, excuse me, that is fine. But I really think you all will do just fine with the lab. So, lab 22, page 110 in your book, just talks about a few things. To successfully follow the dissection instructions, it is essential to be familiar with the following terms. There are four important terms scientists use when they talk about animals. The top one is dorsal. The dorsal is the back or the upper surface of an organism. An easy way to remember that is the dorsal fin of a shark sticks out of the water. Dorsal fin is on his back. Now, your earthworm does not have a fin, but they do have dorsal and ventral, which is their stomach, side. Every animal has a dorsal ventral side. Your ventral side is your stomach. Your dorsal side is your back. Anterior is your head. Antennas are on your head, the anterior side. Posterior is your tail. Those are the four main terms that you are going to need during your dissection and during the live worm portion of the lab. A couple of safety features. When you are dissecting, you are using sharp instruments. I tend to like to use scissors while I am dissecting. I find it much easier than using a scalpel. It is completely up to you. All of your tools you will need for dissecting are in this little blue kit. And we will go over each in a second. But what we're going to do is we are going to skip to part 1 or activity 1. And it is the external features of a live worm. We are going to try to view the physical features and responses that are characteristic of an earthworm. So what I am going to do right now is I have my earthworm over here. Now, unfortunately, I was not able to find a nice big one. So I need to get him out of the dirt. Okay, I have 2 small earthworms that I am going to put on my camera. so you can see them. And hopefully you can see my two earthworms there. One of them is straight and one of them is curled. The curled one definitely is more active. So we have our earthworms. If you have a live earthworm, go ahead and take it out now. If you do not, just look at my worms. Okay, we are going to find or try to find some structures of our earthworm. Now you're going to have to flip back to page 111 while we are doing this in order to identify them. I'm going to use my little probe to point things out to you guys. And I'm going to use my bigger earthworm here. First, I'm going to try to rinse some of the dirt off of him. Okay, now hopefully that will make it easier for you to see him. Now, unfortunately, he obviously is a worm, so he's not going to stay still. But when we are looking at the worm, there is a thickened portion of our worm. Now, on my worm, this thickened portion is right here. It is closer to his head, his anterior end. Here is his posterior end. Now, the only reason I know which is his anterior and his posterior is that thickened area of his body is closer to his anterior or head region. See if I can find it on this worm. Okay, and if you look on this second worm, you can see it over. Well, this one looks like it may be injured. Maybe he got injured at some point. His satellum looks like it's right here. So his anterior, oh, there he goes. Ew, gross. There we go. Okay, and I can hear you guys beeping in, but I actually can't see my screen right now, so you're going to have to be a little bit patient. Okay, so flip to page 114, and on number 1, you are going to label the parts of your worm. So the satellum, or the clitellum, is the thicker part towards the anterior end. So go ahead and label that. And then you can label the anterior end, and you should be able to label your posterior end. These guys are terrific. They are very active, which is great. Now we need to find the setae. The setae are very hard to see unless you have a microscope, and even then they can be very difficult. I will try to hold my magnifying glass so you can maybe see them. The setae, let me grab one of these guys, are little bristles. Yeah, my worms are too small to see this, but they're little bristles along the side of the worm. So you should can label that on page 114. The prostonium, you're also not going to be able to see through my camera lens, but if you have a magnifying glass and you hold it up toward the head end, you may be able to see the prostomium, and it basically is his mouth. I can see it a little, but I'm sure you guys can't see it through the camera. And the very last thing to label is the anus, and the anus is at the worm's back side. All right, so they were both alive, and I'll leave them on the screen for a minute while we make some observations of them. They are quite moving around now. So the questions for this lab are very basic. On page 114, what is the function of the setae? The setae are used to help move the worm. Okay, they're basically little bristles along the outside. Okay, and just for sake of time, we're not going to answer the rest of the questions right now. I just want to make sure we get to the dissection part of the lab. So we are going to see what this worm does, these worms do, in different situations. Okay, if you have an earthworm at home, you're going to take your fingers and just moisten them in a little water, just so that you don't damage the worms. So we have the posterior and the anterior end of our worm. We're going to either take a moist paper towel, or I put the worms on my dissecting tray so they can move around easier. And we're going to see what these worms can do. So the first thing I want you to do is touch the anterior end of your worm. The anterior end is its head. So let me separate my worms here. I'm going to dip my finger in the water, and I'm going to touch the anterior end. And I'm going to see what my worms do. They sort of tend to shrink away from what is touching them. Now I'm going to take my finger and I am going to touch the posterior end of my worm. And when I touch the posterior end of that worm, he tries to get away. Is anyone's worm having a different response than mine? If you do, go ahead and raise your hand and you can share with us. All right, does anyone have a live earthworm at home who would like to do the vinegar part of this lab? The vinegar part of this lab is to take a Q-tip, dip it into some vinegar, and draw a line of vinegar in front of your worm and see what he does. Does anyone want to show us that on camera? Any volunteers? All right, then I'm going to move my camera back and move my worms back. And I'm going to put a vinegar line in front of them. And I'm going to see what they do. Okay, this worm got to my vinegar line and turned away. Let's try it again. Oh, they're both going. My vinegar line is right here. Oh, that worm got in it and is going away. And that worm never got in it, and he is going away as well. Neither of them like the vinegar line. Okay, if your worm did touch the vinegar, which I think this one did, go ahead and rinse it off so he doesn't get hurt by it. If you have your earthworm, or a earthworm, go ahead and pick it up. Of course, I can't get it now. And just see what it feels like and what it does. Okay, mine is flailing around, that one, so let's see what this one does. This one is sort of just hanging out. Okay, and that is where we are going to put our live earthworms away. I have mine in a little cup. I will put them back in my garden later on. All right, so what did your earthworms do? Who is going to share with me? If you had a live earthworm, what did your earthworm do when you picked it up? Anyone want to share? Go ahead and raise your hand. When you picked up your earthworm, what did it do? No one wants to raise their hand? Anyone? Oh, that's right. Not all of you may have had them. Okay, so now we are going to go ahead and get out our earthworms. All right, so a few things before we get started. Everyone needs to have their safety glasses on. If you don't have safety glasses on, go get a pair of sunglasses, something to cover your eyes. Everyone's safety glasses look silly. Doesn't matter. It is meant to protect our eyes. Okay, now you are going to need your dissecting tray. You are going to need your blue kit. You are going to want your magnifying glass. You are going to need your Ziploc of specimens. Inside your box, there's a Ziploc with a worm, an eye, and a crayfish. Take that out, but do not open it yet. And you are going to need some gloves. I am going to go, hold on, and put up a quiz. Once you have everything, go ahead and click yes. Sorry, guys. I'm going to wear gloves while I dissect. I suggest that you do as well. It just will help with the smell on your hands. Okay, so first things first, we need to get Earthworm out, thank you, of his bag. So, you have your bag. Your earthworm is the long worm in there. That is the only worm. Go ahead and take him out and put him right onto your blue dissecting mat. If you have not pulled off the little plastic of your dissecting mat, you are going to need to do that. So take a second, get everything out. And here is my earthworm. Hopefully you can see him. Alright, there is my earthworm. Now I have him kind of twisted so that you can see the whole thing. So, we are on page number 115 of your kit. What you are going to try to do is put its dorsal side up on the dissecting tray. Okay, its dorsal side is its stomach. The stomach of your earthworm is lighter. So I am putting it up. Now sometimes it does not cooperate with you. If it isn't cooperating with you, we will pin it there in a minute. So I'm going to take and open my dissecting tray. In your dissecting tray, you have little pins. I'm not going to actually pin my worm right now. What I'm going to do is take a pin and just kind of wedge it into my mat so that my worm stops rolling over. There we go. Everything that we are going to look at right now is in the top third of our worm. So it's from here to about here. So try to get that part in the middle of your tray. The first tool you will use is your scalpel. Oh, hold on. Your scalpel. If your scalpel does not have a blade like mine, you are going to need to put the blade on the scalpel. Yours probably has a blade on it already. Okay, it just locks in place if you need to put a new blade on. So now I have a blade. You are going to make a small incision to the right of the dorsal blood vessel, which is the dark line that runs along the dorsal side or the belly of your worm. About 1 inch to 1.5 inches posterior, which means behind the clitellum, which is right here. You are trying to cut through the skin and the thin muscle layer because some fluid from the worm's body is going to come out. So before you cut, watch me. I'm going to go about 1 inch to 1.5 inches back. My bloodline is hard to see. And you're going to cut through the muscle and see, if you look closely, I can get a little liquid to come out. That means I have made it both the muscle and the tissue. I'm going to cut a little longer just so you can see what it looks like. You do not need back. You just need to make a little slit. Okay, but when you open it up, that's what you will see. And if you can peel it back a little bit, you have cut through the muscle and the tissue. Go ahead and do that on your worm, please. Okay, I'm going to put a poll up. I want you to click yes when your worm, you've made that initial cut. Okay, now we are going to go a little slow just to make sure everyone stays together for this first dissection. The next tool you are going to use are your scissors. And I need to fix my safety glasses real quick because they came apart. All right. Anthony, once you are there, go ahead and click yes for us. All right, so we are all here now. Now what we are going to do is we are going to take our scissors and we are going to carefully cut using just the very tips of our scissors. We are not going to put them far into our worm. We are going to cut the worm all the way to its head. You may need to turn your dissecting tray so you can get a better angle. Your worm is not all of a sudden going to just flip open. Let me make sure you can see mine and I have a good cutting angle. So I am going to take and I am going to cut using just the tips of my scissors all the way up my worm, all the way to the mouth. Nice and gently, nice and slowly. This is not a race. Go ahead and make your incision. Do not cut your fingers. It is a little tougher to cut through that clitellum. You can reposition your worm as often as you need to. There's nothing wrong with repositioning it. You're going to have to clean your whole tray anyways, so it doesn't matter. Once you're at the tip, you're going to stop. So right now, all you have is just a strip down the center. But in order to see inside of our worm, we need to open it up. So, once your worm is open, go ahead and click yes. Or once your worm is cut. Okay, and remember, we're taking it slow. So once your worm is cut, click yes. Does not have to be open yet, just cut. All right, and once you click yes, just go ahead and give me a thumbs up if you, or a smile face, sorry, if things are going well on your end. A little smile face if they're going well, a frown if this is gross and you never want to do it again. All right, smile faces so far. All right, we've got all smile faces so far, so that's a good sign. Okay, now what we're going to do, and I don't want you to try to worry about building diagrams or anything right now. We're just going to cut and dissect right now. But now we need to open this worm up. So in your lab kit, there are several pins. We're going to use those pins to open up our worm. Watch me first, then you are going to do it. The tray you're using is one of those self-healing blue mats. So you're going to poke the pins right into the mat. Not a big deal at all. So let's get our screen back. And make sure you can see me. Okay. Now you are going to take your pin, starting at the bottom, and you are going to try and pin your worm open. Now your worm has little things inside of it, we said called setae. The setae are inside and out that need to be sort of opened up in order for your worm to get open. So you're going to use the pins to break open these setae. You're sort of using it to detach it. All you need to do to detach it is run the pin right along the side of the skin. And then every once in a while, you are just going to put in a pin. When you are comfortable doing this after watching me, go ahead. Again, the clitellum is going to be the hardest spot for you to work. Doesn't matter how straight your worm is, he can be all twisted. Just want him pinned open so we can see inside of him. Sometimes the setae may be very easy to break. Sometimes it is very difficult. The more times you dissect things, the better you get at pinning. So don't worry if yours is not perfect. Now if you notice a spot where you didn't get through that muscle layer, you can take your scissors back out, like I have a spot right here, and you can just sort of run them along the inside to open it up. As you get closer towards its head, you may need a couple of more pins because that's where there is more stuff. If you have some really stubborn setae, you can use your scissors as well. Okay, so there is my earthworm, opened up. And when you are done, go ahead and click that your worm is open. Robert's a pro at this. His is open already. Sarah's is open already. Awesome job, guys. All right, go ahead and take your gloves off because now we are going to do some labeling. So you can take your gloves off, and if you're careful, you can take your safety glasses off. And we're done dissecting per se. Now we are going to go exploring in our worm. For me, it's easier without the glasses on. Okay, Alicia, once you're done, go ahead and click yes. All right, perfect. So everyone's is open. Now, in a perfect world, your earthworm is going to look very similar. I'm going to turn my camera off to the diagrams up on the whiteboard screen. However, for our first dissection, I am positive that everyone's does not look as perfect as these. We are going to start to try and label our diagrams on page 118 and find different parts of our organism. We are going to start with the digestive system. The digestive system is fairly apparent in an earthworm. Digestive system starts with the mouth, found under the lip-like structure called the prostomium. Then what happens is the food moves through the pharynx and esophagus to the crop. The crop is a soft storage area. It then moves to the gizzard, which is a muscular organ that helps grind the food. The food then moves down the intestines, spanning the length of the worm. Now, we did not cut our whole worm up, so we do not see the entire intestine, but you can see some of it, all the way to the anus, which we did not cut up either. So, looking at your worm and the picture on page 115 and the picture up on the whiteboard, the top one, we are going to try and find some of our important organs. It's on page 115, Anthony, and there's another diagram up in your PowerPoint section at the very top of 115. So the first organ I'm hoping you can find is the mouth. Okay, if you look at your worm, it sort of looks like it's smiling. That is the worm's mouth. Okay, label the mouth on the diagram on page 118. Worms must always be happy because this worm is certainly smiling. But you probably have never looked at the worm, a mouth's worm, before, Alicia, is my guess. A worm's mouth before. All right, now we are going to look at the next thing. The next thing, hopefully, that you can see in your worm is his pharynx. Now, the pharynx of the worm is just inside of the mouth. It is right where you cut, or where your cut ends. If you have cut your satay well, and you have pinned your worm well, you hopefully can see the pharynx. If you need to pin it better or cut all the way through the mouth, now that we've looked at the mouth, go ahead and do so. I'm going to cut through my mouth. I've seen the smile. The pharynx is kind of a whitish, yellowish. Next from the pharynx is the esophagus. Now the esophagus is thinner and looks more like a tube. It runs the whole, not the whole length, it runs part of the worm. And if you separated your satay well, you should see it's like a little, almost like a straw, running down the center of your earthworm. Go ahead and label that on page 118 as well. Then around your esophagus, you should see what looks like little brown horseshoes or black horseshoes. If you see those, go ahead and give me a smile. This is not something we're going to label, but if you see the little black arches, go ahead and give me a smile. Some worms, they're very easy to see. Some, they are not. It all depends on your worm. I can actually see mine fairly well. I'm going to turn my camera on in hopes that I can show you mine. These, let me see if I can do this easily here. Up here, it's not going to work. There are little arches. Those little horseshoes are called aortic arches. Like I said, sometimes you see them very well, sometimes you do not see them well. They are not to be labeled anywhere on your screen or on your diagrams. But my worm had some good ones. Next, for us to label, is the crop. The crop will be after your aortic arches. And it sort of looks like a small, dark balloon. A little like a water balloon. Right underneath it is the gizzard. The gizzard is lighter in color, and if you touch it with your probe, is harder. And the crop is above it and sort of fluffy. It feels like it's full of a liquid. While the crop feels like it's full of, the crop feels like it's full of a liquid. The gizzard feels like it's full of a solid. So go ahead and label them on page 118 and find them in your worm. Okay. You're sort of just labeling on page 118 what it looks like. It is not going to be exact, and that's okay. Some worms are definitely better than others in terms of what you can see in order to dissect. So maybe you got a bad one. The next thing, which you should be able to find, Anthony, is the intestine. The intestine is what is running down the entire center of your worm. So if your mouth is not that clear, move down towards where you started. You are dissecting. Use your probe and look around there. That is the intestine. All right, I assumed the intestine we would all find. And then the anus is all the way at the bottom of your worm. Go ahead and take your probe and just check. Your anus is just a circle. You are correct, Alicia. Probably his last meal. Exactly. So now, that is his digestive system. The animals that we are going to dissect are starting simple, and they are going to get more complex. So each of their systems is going to get more complex as we go. The worm has the simplest digestive system and circulatory system. So let's take a look at his circulatory system. The earthworm's circulatory system is a closed circulatory system, meaning that the blood stays in the vessels throughout the whole body of the worm. The main vessels are the dorsal blood vessel and the ventral blood vessel. The dorsal blood vessel runs up his back, and the ventral blood, tongue twister here, the ventral blood vessel runs up his stomach. almost encircling the esophagus are the aortic arches. Those were the black horseshoes that I showed you. There are 5 of them. In my experience, you're lucky if you can even see one of them. But those are the muscles that pump the blood through the system. If you look on page 119, it's asking you to label the dorsal blood vessels and the aortic arches. Their picture is not a very good one, so don't worry about it. Just see if you can find the dorsal blood vessel. It should be hopefully to the right-hand side of where you cut, but if not, it could be right above where you cut. There should be a little thin black line running up the entire length of your worm. Go ahead and see if you can find it. The next part about our worm is his nervous system. The earthworm's main organ of his nervous system is a mass of nerve tissue called a ganglia, found very close to the prostomium. Now, you can poke around up there and see if you can with your probe. It normally is just a bunch of light-colored material. It looks almost very stringy, and it's pretty well attached to the back or the dorsal side of your worm. So poke around for a minute and see if you can find it. And we're not going to worry about labeling that either, but you can label it on page 119 if you would like. And next you have the reproductive system. The reproductive system of our worm. If you remember from the beginning, I said the earthworm is hermaphroditic. That means it has both male and female sex organs. These are very, very hard to see. If you count from the front of your worm, the testes and ovaries are located between the 10th and 13th segment of your worm. You sometimes can see the seminal vesicles, which is where sperm from the worm that it mates with is stored. They are a large beige structure. I cannot see any of these on my worm. And remember, in terms of our respiratory system, our worm does not have a respiratory system. The earthworm breathes through its skin. The blood goes through the vessels that run close to the skin or the epidermis, and the exchange of gas occurs through the skin. The epidermis is covered with a protective layer called a cuticle that has to be kept moist to allow gases to exchange between the environment and the worm. And that is the end of our dissection. So what I would like all to do is we are going to take a 2 or 3 minute break. I want you to go wash your hands. I want you to take your dissecting tray, the blue mat, and clean it with soap, with a Lysol wipe, and let it start cleaning. If we leave it too long, everything kind of gets stuck around. And any dissecting tools you use are going to clean as well. So we are going to pause for 2 or 3 minutes to go ahead and do that. So I will be back as well. All right, Alicia, are you? If you're back, just go ahead and click OK so that I know everyone is here. And I don't skip out on anybody. All right, awesome. Well, I hope that you enjoyed dissecting. I know for some of you it was not the first time. For some of you it was. It really isn't that bad. It truly is just a minute or two of cutting, followed by some exploring, as I like to tell students. The earthworm is definitely the easiest and simplest dissection we will do. Would anybody like to kind of share? Actually, no. I would like everybody to come up with something to share about the dissection. So, does anyone want to volunteer to go first? Alicia, go ahead.
Hello. Okay, overall, I thought the dissection was pretty cool. I really enjoyed getting to learn about an earthworm. Some of the cutting was a little bit gross, but I really liked it. I liked getting to see the aortic arches. I got a really nice view of those. And the gizzard and the crop, I think, interested me the most. Just the fact that one was more squishy than the other, I guess, or that they varied so much, yet they were right next to each other. And overall, I think it was just a real eye-opening experience, just getting to see, what another animal is like with a scientific purpose. It was really cool.
Awesome. Okay, someone besides Alicia, who's the next volunteer? What did you think there was most interesting, grossest, best part of dissecting? Anthony, go for it.
I thought everything was interesting. The dissection was a little bit tedious, but and I don't think my worm was in the best condition, but it was pretty interesting.
All right, Gabrielle, you're next. Thank you, Anthony.
I thought it was cool to dissect everything because I've never dissected anything before, but it was pretty gross and I didn't like it that much.
All right, Sarah, go ahead. Thank you very much for sharing, Gabrielle.
I think cool to see the dorsal blood vessel. I could see that really well, so that was kind of cool. And the only part that was kind of gross is getting it out of the bag and the box with all the other dead creatures. But it was pretty cool. Yeah, I enjoyed it.
All right. Actually, I kind of think that is sometimes the worst, too, is just getting it out and sort of starting with it. So I agree with you there, Sarah. Okay, Robert, we can give your mic a try. And if not, you can just go ahead and type yours. Nope, that doesn't seem to be working. So why don't you go ahead and type your response, Robert. that door. All right, so as Robert is typing his thoughts, I'm just going to talk about your homework this week. Your homework this week is to do a dissection on your own. What we are going to do is you are going to dissect the crayfish. The crayfish was in the same bag as the earthworm was. You are going to dissect it. I want you to send me at least 3 pictures from the dissection. They can be at any point during your dissection, just showing me the different stages. And then you are going to answer the questions that go along with it. You do not need to write a full and complete lab report. Just go ahead and do the crayfish. If you would like to do the pig, you can do the pig as well. What's going to happen with this class is you are going to be making these dissections. We're actually dissecting every week from now till the end. Anything that we don't use, you are more than welcome to go ahead and do the dissections or the labs on your own. So we will not use the pig, so if you would like to do the pig, you can do the pig as well. But homework for this week is the crayfish. I need 3 pictures at different points, and I need you to go ahead and send me the questions. Anyone have any questions on your homework? I believe the crayfish is the next lab. I believe it's lab number 23. Okay, if you have any questions, I certainly am here to answer them. Otherwise, I hope you guys have a great week and enjoy your crayfish.